Item number 7, we have a presentation from Dr. Madison Trowbridge from Southwest Florida Water Management District, titled Iron, a Limiting Nutrient for Benthic Macroalgae in Florida Springs. The purpose of this presentation is to brief the TAC on macroalgae drivers in the Springs area, provided by spring scientists in the Swift Mud Swim program, to provide further context to discussions on potential macroalgae drivers in Chinook waterways. Connection to the CCMP, this relates to Water Quality Improvement Action 5.1, support harmful algal bloom research and monitoring, and measures to reduce their environmental, social, and economic impacts through the identification of anthropogenic influences. And with that, Madison, you are on.
Thank you, Mark, and thank you all for the invitation. As mentioned, I'm Dr. Madison Trowbridge. I'm the spring scientist at the district here. When I first started this project a few years ago, I never thought I'd see the interest not only in the Springs community, but from water bodies outside of Springs. So I'm very happy to be presenting here today and having been invited for the grant meeting. And some of you may have actually seen this presentation at the UF Water Institute Symposium a few months ago. This presentation today is from a project I refer to as the Iron Project. And I'm going to turn off my camera right now. And if you guys will advance to the next slide. So as Mark mentioned, I work in the surface water improvement and management section, which is known as the swim section in our district. The swim section is arguably the best section because we do get to work on water quality natural systems restoration projects. It is named after the Swim Act of 1987, which directed the water management districts to identify priority water bodies and to establish management plans. We currently have 12 party water bodies within our district, and those water bodies are shown in the slide with the figure on the left. Our district covers 16 counties, all the way from Marion County up in the north to, of course, Charlotte County in the south, and it covers a large portion of the state. In our southern portion of our district, we have, of course, the Charlotte Harbor region, but our Springs areas are primarily up in the northern part of our district, up in Hernando County, which I'm at now, Citrus, and Marion County. We are about an hour or so north of Tampa, and the system I'll be talking about today is actually is our northernmost spring system in our district. Next slide. So before we get talking about this project, I did want to talk a little bit about springs and how they're unique. So springs are, of course, openings in the ground where water flows through them, and they come in many different shapes and sizes. We have some that are very large, some that are very small, some that are circular, and some that look more like a crack on the ground. The springs systems that we work on are often comprised of many different spring vents, and the spring vents all contribute to typically a spring-fed river. These spring-fed rivers often behave differently than some of our other Florida rivers, and this is primarily because of two reasons. The first is that they are the same temperature year-round. The annual mean surface temperature for these spring-fed rivers is about 72 to 74 degrees Fahrenheit. Warm mineral springs in the southern part of our district is an anomaly due to its unique geology, but you'll often hear our springs referred to as warm water refuges, and that's because the spring temperature, that 72, 74, is constant year round. So when it gets those cold snaps that come through, like we had, several weeks ago, and the Gulf water is, say, 50 degrees out, those spring water, I can tell you, feels quite warm. The other factor that makes springs unique from other spring-fed rivers is because they have groundwater as their main source of the riverine input. For some systems, especially for Rainbow River, which we'll be talking about today, we don't see as much surface water input or surface water runoff that's contributing to it. The majority of the water in these rivers is from groundwater. From a management perspective, this means that their direct connection between groundwater and spring water means that we're also looking at management actions within the groundwater. And in addition to that, we also have unique hydrology from each of these systems, especially since they may have different water sources for each of those different vents that I discussed a little bit earlier. Next slide. We have five First Manitou Springs in our district, and First Manitou Springs are the largest springs by the volume of water that flows from them. They discharge over 100 cubic feet per second, which is over 64.6 million gallons per day. It is a lot of water. We have five First Manitou Springs, which include Weeki Wachee, Chaswitzka, Homosassa, Crystal River, Kings Bay, and Rainbow River. And Rainbow River is what we'll be focusing on today. Next slide. Rainbow River is, as I mentioned earlier, in the northern part of our district, located in Marin County near Denellen. The river flows approximately 6 miles from the head springs until it meets the Withlacoochee River at near Denellen. If you have not been to Rainbow River, I do have a little bit of homework for you. It is an amazing system to visit. At the head springs, we have over 250 feet of visibility. The river is home to many different critters. There's a lot of birds, on the water, swimming like araningas, but also, flying around. We also have a lot of otters, which are always really great to see. And Rainbow River also has dense beds of submerged aquatic vegetation, which we call SAV. Next slide. SAV is the benthic vegetation that's present within our spring systems, such as the vegetation that's shown in the photo, which was actually taken in Rainbow River near the head springs. SAV is a critical component of spring-fed rivers, and it has many different ecological functions. For example, it helps to stabilize sediments, it uptakes nutrients, it also provides food and habitat for many different organisms. I do want to note that SAV is not the same as seagrass. So seagrasses, which you all may be more familiar with, are what we consider halophytes, so they need salt to live. However, the SAV in our springs are not very salt tolerant. Some of our coastal springs do have vegetation that is a little bit more salt tolerant, but generally these are freshwater species. Next slide. SAB in our springs can be categorized into one of three types. We have our desirables, our undesirables, and our algae. The desirable vegetation is essentially our native vegetation. So in a lot of our springs, especially in Rainbow River, we can see these dense beds of SAB, which you can see as in the photo. And some of these uh, vegetation can get up to 6 feet long, primarily up in the upper river of Rainbow River. We also have invasive species. These were, of course, species that were non-native to Florida that were eventually introduced over time, such as the hydrilla, which we see primarily in the lower part of Rainbow River. And then we also have algae, which is also prevalent in the lower portion of Rainbow River. Next slide. When we're talking about our algae or macroalgae, there's pretty much two main types that we're talking about in Rainbow River. The first type is what our focus will be on today, which is the benthic algae. So the benthic algae is just going to be the algae that's right on the sediments. And this species is primarily Lyngbia within the Rainbow River system. We also have epiphytic algae, which is, of course, what will blanket our vegetation. And within the Rainbow River system, we see this primarily as Spirogyra species. Although algae is technically considered as a native species, the excess of this algae can indicate an ecological imbalance within our river systems, which is why it's often considered a nuisance or undesirable. Next slide. This imbalance has often been attributed to nitrate eutrophication in our spring systems. So over the past few decades, we've seen elevated nitrates in our spring water through a variety of different sources, primarily fertilizers, septic tanks, and agriculture, contributing to nitrates to our groundwater, which eventually comes out of our springs. The nitrate has been, this excess nitrate, I should say, has been attributed to excess algae. And this is because algae is unicellular and it can uptake nutrients and grow faster than our rooted vegetation. Eventually, this can lead to smothering of the vegetation due to the lack of light, which leads to an algal-dominated community. In the past decade or so, there's been an increasing body of evidence within the literature that has not found the correlations between nitrate and algal abundance within our spring systems. Instead, we've seen more correlations to other variables such as flow velocity, dissolved oxygen, and even in reductions of the amount of invertebrates that eat algae. Next slide. The district has also recognized that something unique was going on in the Rainbow River system because it does not follow that nitrogen paradigm that we just saw. Rainbow River has some of the highest nitrate concentrations of all of our first magnitude spring systems. It sits about just under 3 milligrams per liter. But it also has some of the healthiest SAV in the head springs. Some in the head springs area, we can see 100% coverage with 6 foot long blades within that area. It's only in the lower portion of Rainbow River, closer to the Withlacoochee River, where nitrogen is actually lower, that we are seeing the algae present. So the district took a deeper dive, pun intended, into the dynamics of the Rainbow River spring system. Next slide. So this work culminated in the Hensley et al. 2017 report. This is a multi-part, over 200-page analysis on the Rainbow River system. It's really an in-depth analysis, but I'm going to distill a couple of the major takeaways from this report for you all. One of the things that this report did was it utilized what we refer to as these benthic chambers, which you can see a photo of on with the photo on the right. The benthic chambers essentially were used to test metabolism. So there would be nutrients that were added to these benthic chambers, and then the response of the SAB or the algae was tested. The report had three main findings. The first was that the uh, gross primary production, known as GPP, which is essentially just the rate of plant metabolism in the plant community, was not limited by nitrate, and based on their analysis, potentially never was in the Rainbow River system. And they also found that filamentous algal coverage was not correlated to water velocity, which, based on the literature that was available at the time, that was the kind of more prevalent hypotheses as to what could be the reason for the algae in the lower river. What they did find was that primary production and the algal biomass were stimulated by the addition of iron to these benthic chambers. Next slide. This leads us to the iron project, which is essentially just testing the relationships between algae and iron. And this is a follow-up on the Hensley et al. 2017 study. So what we did is we took over 60 samples throughout Rainbow River, Head Springs middle and the lower portions. And we looked at areas that had algae present at varying different coverages, and also what I refer to as control areas, so areas that did not have algae present, typically more of the bare sand areas. We did this over two sampling periods, one in kind of our winter time and one in our summertime, because this is when we could potentially see seasonality within our spring systems. So we were looking to see if there was any seasonality, and we didn't see that for this report. What we looked at was the benthic algal coverage as well as the thickness. So essentially just the percent coverage in a quadrat, as well as how thick it was, essentially just measuring with a ruler how deep that algae extended from the bottom. We tested both pore water and water column chemistry. For the water column, as you can see in the figure on the right, we did the water just above the algae, and the poured water we collected through, basically it looked like a needle that was pulled up through a series of tubes. Next slide. So let's talk about the findings from this study. The first thing that we found was that there was more iron in the sediments than in the water column, and this wasn't a surprise. We knew that there was going to be more iron in the sediments just because sediments have, well, especially the clays that we can find in some of our spring systems, have a higher affinity for iron than our spring water. And spring water generally, it does not have a lot of iron present within it. We also found that within the sediments, while there were patches of or pockets, I should say, of areas that had a lot of iron present, the lower portion of the river tended to have more areas that had iron present within them. And that makes sense based on the local knowledge about the Rainbow River. There's a unique history with the lower river, as well as it has a unique geomorphology in this area. And this area often has more clay sediments, as I mentioned. Next slide. We did find a correlation between algae and pore water iron. This was very exciting, and I wanted to share a story from the field which relates to the picture on the right. There was an area near the head springs that had this pocket of algae around it, or pocket of algae, and there was a bunch of submerged vegetation, the rooted vegetation around it. And when we were sampling the pore water, you can see the results of that with the figure on, or the photo on the right. And that was that we were seeing that there was a lot of iron, as you can see, because the sampling tube turned bright orange. And it was really interesting to see that what we were finding in the field, where there's these pockets or patches of algae, where we were seeing these pockets of iron, and we were able to corroborate that with our statistical analyses. Next slide. So, of course, that begs the question, why iron? Of all things, why iron? Well, iron is a critical component to all life, and SAB is no exception to that. However, algae need more iron than native vegetation, so that the rooted vegetation needs less iron than algae. And this difference is about a tenfold difference. So, and we also know that through various mechanisms, both geochemical, physical, as well as biological mechanisms, algae and plants can uptake nutrients like iron from the sediments. So areas where there is elevated iron would allow for algae to have the competitive advantage, and vice versa. Areas where there's less iron therefore give the SAV that competitive advantage. Next slide. This is not to discount the role of nitrogen within our springs. Nitrate reduction is still a very important management action in our spring systems. Uh, though the exact mechanism is not understood, there's some evidence in the literature that suggests that nitrate eutrophication may be exacerbating the iron limitation, or could cause iron limitation within these SAV, both the rooted vegetation as well as the algae. It's also important to note that our spring systems can literally have downstream impacts. So, for example, the Rainbow River system flows to the Withlacoochee River, our coastal springs flow to the Gulf of Mexico. So, it's important to recognize that the reduction of nitrogen in these spring systems can benefit the receiving waters. And interestingly, when we're looking through the some of the literature out there, the there is evidence that suggests that nitrate eutrophication may actually be negatively impacting the rooted vegetation in our springs. So, yes, of course, nitrate reduction is still a very viable management action, but it's the reduction of nitrates alone may not help us achieve our goals in our spring systems. And we knew that going into that. There's been a lot of literature out there that supports those findings. But with this work, what we see is that iron may be more of a limiting nutrient, whereas iron may be more of a limiting nutrient, and nitrate may be more of an inhibitory agent within the SAB of our springs. And by looking at it in this light, iron really seemed to be that missing piece of the puzzle that we were looking at when we were looking at the springs literature. So naturally, when we finished this project, we had more questions than we had answers. So we are doing some additional work. We have a couple ongoing and upcoming projects with this. The first, we're trying to understand, is this a unique phenomenon to just the Rainbow River system, or is it occurring in some of, say, our coastal spring systems? And we're also trying to understand if this is unique to the benthic algae, or if within the Rainbow River system, or if it's also occurring with epiphytic algae. So next slide. In regards to Charlotte Harbor, I'm not certain to what extent iron may be playing a role with the marine algae as far as your discussions, but I did want to present this work kind of as a lessons learned, and we are still exploring some of these aspects. Um, we are finding that sediments are um, more impactful than we had originally thought. And I think as a scientific community, we do place a lot of emphasis on the water column, and I think that's wonderful. I think that's great. We have a very robust water quality monitoring network, which is amazing. But I wanted to share this information in case there are areas that may benefit from a targeted approach. And by looking at the sediments, you may have an additional tool in your tool belt. With that, I do want to thank you all for your time, attention, and the invitation to speak here. You can scan the QR code to sign up to our newsletter to find out more that we're about what we're doing to protect our springs. And I think we have time for questions.
Thanks, Madison. Great presentation. We have plenty of time for questions, so yeah, don't hold back.
God, I got a question. I'm sorry, I got it.
Yeah,
Is the iron, of course, is coming from the ground, right? It's has been located places where it's more concentrated than others in the river. You have this like a graph where it has a different concentration, but there's a source of that iron in a specific location, like a underground mine.
That's a great question. With the history of the Rainbow River system, we do see some unique geomorphology within the river. There are some areas that tend to just have more of those clay sediments, and when we're looking at the pore water, I don't know to what extent that it's coming necessarily, from deeper within. But one of the things that we're investigating as for the kind of upcoming study that we're looking at is there's a spring vent that is near an area where we're seeing some localized algal blooms, and so we're trying to see if maybe there's that groundwater surface water connection that I talked about. We don't have any information on that yet, but it's something that's definitely one of those questions that we have that we're trying to investigate.
Thank you.
Mark, I've got one.
Yeah, from Mark.
Hey, Madison, this is Mark Schrammick. I work with the National Marine Fisheries Service, and I do a fair bit of conservation work in the lower Homosassa as well as Crystal River estuaries. And I appreciate the work and the input you've done. I'm just kind of curious, in the Rainbow River, particularly up in the headwaters, do you see any Valsenaria americana, or is it almost exclusively Sagittaria, I think you had said?
Rainbow River is really cool because at the head springs we see Sagittaria curziana, and then it transitions about KP Hole to more Vallisneria americana, and we get a mix of both within the river. So sometimes some areas we'll see some more Sagittaria, some areas we see more Vallisneria. There's It's not really well understood why within the literature where you get to see those changes. For talking with the St. John's Water Management District, in both the Silver and the Rainbow Rivers, there are times when the Vallisneria and the Sagittaria, phenotypically look very similar. You actually have to touch them to be able to distinguish the difference. So we do see both within the system.
All right, good to know. And clearly it's not, KP Hole is definitely not tidally influenced. So I was just curious if there was a transition. It sounds like there is one, but any correlation as to why they might occur where they do?
There has been some, when you look at the literature, there's some ideas as to what it could be. Some of it may be, we're not sure if it's light, if it's flow, if it's maybe uh, sediment related. We know that Vallisneria is very well adapted to a lot of different types or has a broader range of what it can live in versus Sagittaria. We're just not 100% sure what parameter or what combination of parameters it is at this time. And I'd be happy if you reach out to me. My email's there. I can send you that paper that was published by Rob Matson from St. John's on that.
Cool. Thank you, Madison.
Any other questions? Well, I think I'll add a comment, I guess, just to echo Madison's final points. I think that one of the big value of this work is it challenges us all to think a little bit outside our box. We do sort of get used to looking at the problems of eutrophication through one light, and it reminds us that these systems are complex, and perhaps that when we get into our CCMP update, when we look at things like numeric nutrient criteria, it's worth keeping in mind that one particular nutrient may not be telling the whole story. So while we're out there, it's definitely worth looking at those greater connections between different elements and different parts of the system, and also between the sediments as well. So Madison, thank you very much for bringing this to the TAC, and great work.
Thank you.